Hey guys.
Yeah,
Hello.
I'll give us a couple minutes. We'll meander about.
That sounds good. I'm going half workshop Q&A style, so I plan to make it informative but conversational.
Gotcha.
We'll get
Okay,
people
guys.
flowing in.
Yeah, I'll give us a couple minutes.
We're gonna have a live stream running through the Brain STEM Center concurrently, which I've decided isn't going to necessarily help this group here, but that is available if anybody somehow happens to encounter it on YouTube. But if you're joining the Zoom link, you have come to the right place so far. So hopefully we have people file in here. Yeah, I'll give them a couple minutes over at Moss. Looks like we've got a few people remoting in from elsewhere.
You're gonna lose some.
Okay.
Okay. All right. Go ahead and try it now, and then we can. Is the cable defective?
The last one was just kind of loose. Like whenever we wiggled it. Okay, this one seems. And now we're gonna want to test sound because I don't know where it's gonna come from. Your mic's still your laptop. That's probably the projector.
Oh, is that going to broadcast like if audience from elsewhere is talking? Can we have somebody unmute and just say a couple words on your end, and we'll see if folks can hear it here?
Testing. Anything to
All right, I've got you. I guess it's not a huge deal if it won't come through there. We can just kind of I'll relay what they say.
Yeah.
Otherwise, we're going to troubleshoot this mic for 10 minutes. And,
Yeah.
yeah. Okay. All right. So we're probably good there. Um,
They can still hear you.
yeah, I'm going to get my live stream out of the way. Okay.
And then you just need to click record.
Remind me to click that in a sec. I'm going to wait till we get underway. I think I'll do local.
Yeah.
I don't know why. I just worry that the cloud is going to be unstable at some point. Yeah, we're not doing that though. So just nudge me after this and I'll share the file.
Or do you want to share the brainstem?
It is on Brainstem.
So like go to the channel, live, share, and copy that.
Did I just copy this one?
Maybe
Well, this.
no, go to on the right,
Where?
the pen brainstem symbol. At the very top.
Oh,
Yeah, there
thank you.
go. Channel, like your view your channel or whatever, the blue thing. Yeah, there you go. And then live.
What do I do?
And sorry, go to the live. And then bottom. Yeah, those three dots on the side. Share.
All right. Want me to send that to you and you can circulate?
Yes.
Thank you. All right, I just sent it to you. I'm gonna kind of get us underway then. Oh, hi, Branch.
Fine, afternoon.
Thanks for joining. All right, can I, how are we doing on the Moss end? Are you close to a critical mass there?
I think pretty much everyone who's going to be in person is here.
Cool.
Yeah, I know Laurel's not in today, so she won't be attending.
All right. Yeah, well,
I don't
and
know. I was gonna say, I don't know where all the post are.
okay. Well, somebody go, flagellate them for this crucial educational experience. And yeah, so why don't I just kind of do our quick preamble and get us moving. So thanks again to Laurel and Branch for nudging this opportunity to happen. I would like to think of this as the start of a longer community formulating exercise. So I'm hoping I get to know some of you who I don't know as well a little better through these interactions. I think the spirit of this from my vantage point is having previously been a T32 mentee and focused a lot of my career on imaging and network science and computation. is I still find the interesting problem is how does this relate to an advanced neuroscience and make patients' lives better? So this will be quite a bit of didactic from work we've done, but you're going to get a bit of philosophy on the front end, a lot about sort of the foundation biophysically about where people think structural connectivity comes from. And I'm going to use our own work over the last couple of years as, I think, on the one hand, good examples of certain network concepts, but places where we really have a lot of room to grow in terms of advancing the sort of neurophysiology and neurorehab questions. So that's where I'm coming from to this talk. Let me get the talk itself booted up here. And yeah, I'll record as soon as I get the, okay, all right, cool. So let me get our record tool and then I'll be able to share that with whoever couldn't make it. All right, you're now all on watch, so be warned, we'll all be on our best behavior now. So if you don't know where I am physically, so I'm currently at Stratton Hall on Drexel's campus. I'm just a stone's throw from the edge of Penn's campus. And so I'm then just across town from some of you in the audience. And I have a network of very fortunate collaborations at this point within Drexel, over at Penn and LCNS and Brainstem, and Moss for Rehab, and therefore now Jefferson. And when I was putting this series together, we first started with a brief primer on some connectome preprocessing with the Porvik Elkar, who's been really the only reason a lot of the work gets started in the first place. I still have the view that to do a competent preprocessing and imaging only gets more sophisticated every passing year. We have the big advantage of Penn's BBL laboratory and their XCP engine. So if you missed that talk and just want really just kind of a blitz overview of how that's structured, I recommend hers. That should be shared up on the Moss and ultimately Brennan STEM sites by the end of this series. Where I'm picking up is here, structural connectivity, which is what we're going to discuss today. That's going to give a foundation for functional connectivity analysis. You'll start to get a sense today for how I really think of these as intimately related companions. So there really is a 3-act play here, where by the end, I want to talk about why are we treating all of these elements separately, and how can we think more creatively and productively about how they relate together. So I'm an academic. This is going to feel a little bit like a talk at first, but I'm going to build in a couple Q&A points. If you don't stop me midway, please also stop me midway. And so we'll go around 90 minutes or so, or whenever it's interesting to folks, and then pick it up in a couple weeks. Sound good? All right, awesome. And thank you all for being here, because for me, I think a lot about this question, which is how do you ask a good network neuroscience question? And I think there are a few ways to answer that question, but I'm going to give you one that I think really has some deep foundations, not just in analysis, but literally in the philosophy of neuroscience. And so I think in a nutshell, if you're thinking about networks or connectivity, I really enjoy and recommend this paper by Rick Betzel, who worked with Danny Bassett part of the same time when I was in her lab. And the argument is really, it gives you a fundamentally new dimension of data analysis. So we often, when we come up as undergrads and grad students, think about the brain in space. You know, maybe you're interested in whole big brain maps like I tend to look at. Maybe you're looking at the cellular level, somewhere in between. You've got a temporal dimension. I tend to increasingly think of what's happening right now and what I can do about it, but you might be interested in what happens over days and years and lifespans. But really what network analysis is doing for you is adding a topological dimension. It's saying that, yes, it involves bits of space and time. That's, you know, we're still working within physics for the brain, we think. But what you're really saying is that this is more than just 2 dimensions. This, in fact, is very many dimensions interacting. These are the pieces of the brain. These are the nodes and the edges. And so the problem in this space, if you're new, is it's endless, overwhelming, ever-growing, and only broadening and quickening. And so I think that on the one hand, you can say, that's too much, it's overwhelming, I don't know how to get started. You can say, oh my God, I need a new PhD and go and do that process. Those are sometimes good decisions. What I want to do today in our community is point out, I really do think there's a way at this point to reduce how you ask questions to make them more meaningful at the first time you ask them. And that therefore constrains how you ask them in your data. That makes progress theoretically a little more meaningful. And I'm going to explain how I think about that. So let's return to this. What is a good network neuroscience question? Well, I don't think there's anything special in a lot in science, right? We've had conversations about this for hundreds of years, it turns out. And most philosophers of science agree that it can occur in one of three forms. Enlighten it about any topic, including the brain. You can find some kind of relationship with other known variables. You probably know this as correlational analysis, analysis of variance, things like that. You can think about principles. I tend to think about this as causes and effects that govern phenomena, so adding some directedness to your claims. Or you can find new things that you couldn't interpret and or you maybe couldn't explain before by the prior knowledge that we have. I think when you take that general framework and ask your neuroscience questions with networks, you've really got things that happen in three categories. One is that you add measures for verbal or visual concepts in neuroscience. So for instance, one thing I'm doing with some of the colleagues on the call is there's something like a hub theory of semantic processing in the brain. Turns out people use words like hubs without often adding mathematics or real definitions to what you would look for in the data. And so I'm interested in that problem in general, which is when is it loose associations? This doesn't mean anything, or when is this a good measure for your question? So that's the first thing. The second thing is you found a new thing, right? So we add an entire dimension I'm calling topology, the way things are organized in a network. And there's really a happy hunting ground there still, I think, in network analysis and neuroscience. I think for long past my lifetime, we'll continue to find new ways of articulating network structures and functions, and that's a descriptive process. What is this thing, and how can I describe it? The third thing, and what I personally am the most interested in, are things that test, contribute to, revise, or refute existing domain models. And to me, this is about mechanisms. So my favorite little infographic from a paper we published almost a decade ago is, how do you map this problem from here on the left, something like Brodmann's map, cells configured in different ways, at a low level into bigger groups that define how the network is organized and how maybe it functions together. And I'm going to make the argument today that structure really is a potent foundation of that. And how do you go about then doing meaningful analysis in that space? Does that make sense to everybody so far? Okay. All right. So this is sort of the common way I've seen things go. And I'm going to give you two scenarios because I believe in science that how we talk to each other says a lot about how well we do. And I might turn out to be wrong about that, but so far here's one scenario. You often have somebody who I would call a domain neuroscientist. You know something about the brain. And I think it's okay to admit we know some things about the brain, as you'll see. And so we'll come along and I'll say I'm interested in motor systems or semantics or semantic memory or attention, whatever it is. And you meet up maybe and say, I think a network is interesting. Brains are networks. This is an interesting network problem. So this is the handoff problem, and what I would argue we want to avoid when we do network analysis. So the handoff problem looks something like this. The domain neuroscientist said, I think I have a question that might need a network method. The network scientist says something like, all right, well, here's an option for that based on what I'm hearing, but it doesn't really have any fundamental principle in common with that domain question. You'll see a couple examples of this as I talk later on. But basically, we have some words, we think we mean the same thing, but then we find out later we don't mean the same thing. And so the first problem is about communication and semantics. What usually happens in this case is the domain neuroscientist then gets back a bunch of network stuff, and they say, if they're being honest, I don't really understand it, and they can't easily relate it to the content domain. And then what will often happen in that case is that the network scientist will look to explanations within network theory to then apply to that knowledge. And the liability in this case is maybe you actually did find something new, but you can't figure out what the significant is in one or the other or both domains. So we kind of follow what's wrong in that scenario. I don't know the commonality of this, but I have lived this, I have seen this, and I'm kind of now making it an academic project to get better at avoiding this in the field. And I think this is really a shame because what this means is we didn't spend a little bit more time at the first step trying to figure out what we meant by networking connectivity and talk that through. And I'm really giving you my internal model for how I've updated that prior at this stage in my career. So what's a better way to do it? I'd say it's the collaborative solution. So I think true collaboration in network data analysis is you figure out how to ask a question that requires network methods. You'll see a little bit about how we can notice the difference, I think, today. But what I mean by requires network methods is you simply couldn't have couldn't have done your analysis without articulating it as something like a graph theoretical construct, something that would be expressed in a mathematical formalism that a network theorist would recommend. Not all questions function that way. I think it's important to say. So identifying the difference is really important. So the network scientist, when hearing it posed this way, might be faster and better at saying, oh, that sounds like this kind of network analysis or measure. Well, now we're cooking because both of us are actually semantically talking about similar things. We get excited about the commonality. We figure out if we're doing something new and interesting, we go for it. We get some data back. The domain scientist looks at it and says, oh, like, that's pretty close to what I thought we were talking about, but it's a little bit different in this way. Maybe we need to kind of go back around and tweak it and interpret it differently or do a supplemental analysis. That requires, I would argue, context. This is the person who's been training in this probably for many years, probably knows a lot more than nothing about it, and is going to have a very keen eye to like, that's just not adding up, and I can point over here and tell you why, and you want to have that interaction. And then finally, I think you adapt to this as many times as it takes in some cases, where you tailor then your analysis to meet the demands of that question. And usually when it's a good network question, that's when you can bring in the domain general knowledge about networks and say, we know this in general of networks in other cases. We also see it in the brain. Maybe there's some correspondence there. Those are usually the most robust and meaningful outcomes where we found something new. We have some scientific value that all the authors agree about, and the follow-up questions can be posed, usually by those very authors. So my best advice, I struggled to think about this. I got anxious about this talk, really thinking, what did I want to say to kick off the next two talks? And oh, yes, please. Oh, is that just a cough from the audience? All right, I'll go ahead. Stop me if it was a cough. It wasn't a cough. All right, so I think, unless you really think we haven't learned anything from 100 or more or 150 years in neuroscience, please don't forget what you thought you knew about brains when you start to think about a network study. And you're going to kind of see my general and self-critique of our own recent work is often an error in this problem. It's kind of looking around and saying, like, we did that, and it's kind of cool in these various ways, but it's missing some of these foundational knowledge, pieces of knowledge, and we didn't test these assumptions. And I'll show you a few instances of that. So look back at your neuroanatomy. We're going to talk mostly about things like on the left, where we know something about structure. And I think know with a capital K scientifically, like we're pretty certain about certain things. And we want to think about other levels like computation here from Cajal and things like regional heterogeneity, Brodmann. And again, we've known these things for more than 100 years. So I really want to challenge us though today to say, what does a network analysis add? When is a kind of a sniff test for when you need something like a connectivity or network analysis? Well, I think we're going to start on the left talking about node features or paths. And so I want to anchor most of what I say just because this is where I've done the lion's share of this stuff recently, close to examples in language and aphasia. Most of you probably encountered in a textbook or in a patient setting the idea of Broca's area and Wernicke's area and the communicating arcuate fasciculus between the two. And you can break down aphasias sort of coarsely into if you're lesioned in one place or the other, the connection, right? So that's kind of familiar probably to most people. So great. I would say we still are learning things about that approach to it, and we should continue to learn them. When you might need a network question is when you realize that, well, not only am I interested in Broca's and Wernicke's and that connection, I'm starting to worry about the connections with other things around them. So if for any reason, I think that the interactions between Broca's area and other parts of the system might matter, I might need something that can measure that thing about the system. And we'll talk about a couple instances today. But you might have heard of concepts like node degree or node strength. These are basically just kind of counting up other connections around that node. Maybe you filtered that by you think 5 or 6 things matter. Maybe you've said, well, maybe about the way everything inputs into that node matters. Let's investigate. I'll give you examples. Paths are very interesting. Paths are now analyzed in a lot of contexts now, but basically this is the idea that Wernicke's and Broca's area and the arcuate are part of a longer chain of processing in the brain. And so if your circuit doesn't end at just one end and the other, you're going to need to add other links in the chain. So paths or walks and other measures related to that are usually, well, let's add some steps along the chain, and we're going to build some measures for them. Okay. Now, what you may have noticed is exploding in the literature alongside these node and path measures is the idea of doing whole brain analyses of networks and then trying to make associations between large scale measures and either large or specific behaviors. So there's again, no way to say all of these in a single talk and also do anything else. So I'm just going to go after a couple of main concepts you'll see over and over at this point. So the idea of modularity is, you can see here 3 different colors on the map. You can hopefully appreciate that there are little groups of those blue nodes in those modules. And then you're going to notice immediately that you can count modules, and you can also find stuff in between the modules, connecting those modules. So some of the nodes in the brain are going to work kind of locally within big groups. Others are going to mediate here between groups. And there are various measures for that. Participation coefficient, people have called these, local hubs and connector hubs, and you'll scan for various things in the literature about that. The concept of modularity is interesting and old. This predates network analysis in the formal sense of just what is a modular mind. And you can talk about modularity in function and concept space, like how do concepts chunk together in a sort of abstract sense, or you can ask, how is the brain itself modular? And do those things relate to each other or not? And so there are strains of literature focused on those questions. They continue to develop. Modularity has become interesting in stroke literature. I'm going to talk at some length about that. We're doing a new large-scale simulation that I can mention at the end to ask about, well, when I start to break specific pieces of the elements of the system, these are your local parts, meaning what if I blow out this node or this edge or groups of those things? Notice that as soon as I do that, other things are going to break too. And so the thing I find fascinating about the brain is we spend a lot of time, including in imaging analysis, sort of trying to isolate down to like, here's the variable and its relationship with my outcome. But what we don't often stop to think about and test is, well, when one thing changes, by definition, in a network sense, something else did too. And we can see that over and over again. But I would actually say one of the areas that I've noticed is missing in the literature, especially in the last couple of years, is explicit tests for those links. What shifts together? What moves together in the network? What by definition else changes about a network when I break this little thing? What varies across people? These are questions we can talk about. Small worldness is another domain general network idea. And the idea here is that, well, how would you build a network that's really good at doing lots of things? Certainly it should be somewhat modular. It's got to have some specialization in organization. That's a kind of efficiency. But there's also efficiency in paths. So our brains, it turns out, are very efficiently wired up physically, structurally, such that you can get anywhere else in that network within just a small handful of hops. And you've probably seen this in the game, degrees of separation to Kevin Bacon. And it turns out that path length through the network shrinks in social networks, and it's also quite small in brain networks. So in this handful of hops, if you can get anywhere, you also want to worry about, well, getting anywhere could be cool, but there's also some local structure, as we mentioned. And so usually this idea is paired with clustering. This little purple triangle is saying there's little neighborhoods of things that really tightly couple, but you can get to any neighborhood in the brain very quickly. This works in highway networks. This works in social networks. This works also in brains. And we'll mention more about that. So these are examples of what network analysis adds. So let's talk a little bit about, I think from the ground up, what that means physically for structural networks. And then, well, I'll share through some of our own work examples, and I'll insist we have a Q&A if I don't get stopped at some point in the meantime. All right, so why am I doing this? If you've noticed anything from our lab, we've put several connectivity structural papers out, and we haven't included the function yet. I think the second thing is a problem, which we'll mention in the last talk of the series. But why am I suddenly insisting we spend some time on this? Most of your brain is white matter. And I think when you look at a brain and kind of strip away the gray matter we usually start out with in textbooks, I appreciate better every passing quarter just what complexity we don't understand about the way things are wired up and how that mediates communication. So by volume, it's somewhere around or more than 90%. A lot of your brain is usually talked about in terms of these associated and long communicating fibers. That's what the atlases will show you. The percentage of mass is mostly made up by U fibers, for which we don't have good atlases or tractography for. So we'll kind of return to that theme a bit later. But where we're at right now is we have this wonderful imaging technology. It's diffusion imaging. It comes in several forms. I'll mention a few. And once you have this technology, you can make inferences based on kind of the average water flow of molecules and then build these tractograms. And if you look at them, like meaning you actually run the analysis and look at it, you're going to see, if you peer at it, things like U fibers and communicating fibers, and they're constrained in the brain, and you're getting at the white matter. And we'll talk about how well in a minute. Network analysis comes in when we start to say, well, let's reduce that into a problem of networks. We're going to say there are spots on the network, there are connections between them. Now I can ask some questions about that. So the reason that I think we need to spend time on structural connectivity is not too dissimilar from the way roads work. Your brain, not always, but for the most part, communicates a lot of its activity through white matter. A large mass of communication comes through that, and very much like the constraints of roadways across the United States. Brain signals, by analogy, are something like traffic. It changes its rates, it changes its format, it changes its content, but usually the cars are on the road, right? You're going to get a few of them going cross country over the desert, and one of them's going to flip over the lane when somebody's a little reckless. Like, brains are kind of messy too, but we can kind of walk away with a general intuition like that. So if we accept that the structure in its pathways has something to do with the way that it works, why would we study that in clinical samples? Well, so I think some of that is entirely just kind of the annals of academia, where you describe some features, it's kind of cool. Maybe you get to name it if you discover something new and you feel good about yourself, right? When we're talking about it in terms of diagnosis and connectivity analysis, this is the really interesting part of the problem to myself. Most of you at some point have probably seen an image like on the left, where you have some sort of lost volume due to a lesion. You have some kind of swelling and a cascade of effects, and eventually it'll probably stabilize if it's stroke, more or less, to have a large mass loss from that. If you're to go do a diffusion imaging, this is from a real patient example, you're going to kind of see something that at first glance might be totally unremarkable to you. You'll be like, yep, I saw a lesion there, it's knocking it out. You might, if you're looking close, already guesstimate the arcuate is probably gone from this person. They're going to have some language problems. Big MCA, infarct, here's a problem, right? And so this would be sort of like, what else have I learned than what we already thought we did in the textbook about Broca's and Wernicke's and communicating fibers? But here's really what happens. What really happens when you do something that looks like kind of a lesion, this focal-ish mass, is because it's intersected the white matter, usually you have a tremendous effect on how all of the pathways are affected in that hemisphere. What I mean by all of the pathways is that principle I mentioned earlier, which is if I change something here, what I measure about over there, even in the structure, may now be different because I've modified that entire highway structure, right? So here on the top right, we have the image from the same patient where what might have been kind of like pulling your eye toward, oh, I'm thinking about classic language phenomena. Really what you're talking about is massive generalized trauma to the left hemisphere, which, by the way, is connected to the right hemisphere in normal circumstances. And now the whole network has changed in some way that you may not know until you measure it. And that is where I think the interesting set of potential questions takes off. What do we want to know about what happened on the right that says more than the image here on the bottom left? What is actually novel and interesting here, and not just some kind of messy set of correlations in noisy data? Okay. So once we notice that this happens on the top right, hopefully it becomes apparent very quickly that maybe that modular structure also shifted, right? Maybe I bisected one of these connector nodes directly. Maybe I didn't. Maybe the difference is important for my outcomes, and so on. We can start to ask about, how does this sort of macro structure shift with what I thought was a local effect? What is global versus what is local? So when we do this in connectomes, let me just define some data and elements. We tend to take something like the tractography on the left. We've done our water diffusion tractography. We get some kind of measure. Here's a matrix, which is called an adjacency matrix. So we're asking how close by are things, and you can load in all kinds of measures, and I'll mention a few later. But here we tend to organize it in modules. We usually have a flipped dimension here, so that's why right is left and left is right. But basically, you kind of come down this matrix. It's bidirectional because it's structural connectivity. You're going to see a lot of colored elements, which are just a measure of how strongly connected is that region pair, right? So if I have more integrity in that tract or more directedness, I get a higher value. Great. This is what you expect to see. You're going to see stuff connected within hemispheres. That's where a lot of the connections are. You're going to see some things that start to look like squares. Those will be detected as modules if you run an algorithm on them. You're going to see some stuff communicating between the hemispheres. A lot of that goes through the corpus callosum. And if you took both hemispheres and kind of squint at it blurry, they're pretty similar in most normal people most of the time, with some interesting small differences. Now let's say I have something egregious like a stroke happen to this poor person, and we're going to have this apparent effect like we saw in the tractography if you actually just open it up in the software. But if you actually compare this matrix to a relevant control and just kind of, mark out where did I lose an edge, usually you can see there's a profound loss of edges, even for relatively small strokes. And so a lot by volume of that connectome is directly disturbed by the stroke, and the area around that stroke and those lost connections may also be modified due to the mathematics of how the network is wired up. So once we've done this game, we've defined a parcellation, we've pulled out these connections, we have the mathematical object. Well, now the fun begins, and here's where also the danger can occur, right? Because we can easily get excited and confused and distracted, but we can also learn a lot. So let's talk a little bit about that. These are the decisions I think everybody makes, even if they didn't realize they made these decisions, okay? So I think there's a lot of sort of just momentum in certain scientific fields. Network science is no exception to this. You don't get away from realizing whether you had a hypothesis or it's data-driven. Sometimes when you do something that you think is data-driven, you had a secret hypothesis you were hoping is true. Sometimes when you did something hypothesis-driven, you said, whoops, that didn't work out. How am I going to get my grant funded? And then you go on a wild expedition looking for new things, right? I'm just going to assume everybody's familiar with this phenomenon I'm describing, right? When you get to brass tacks and you say, yeah, but I really want to study it, you have to define regions. And I think that that's some people have offered alternatives to even trying to make that decision, but by and large, this is what you do when you do a structural analysis. You say, what's the set of regions that I'm starting with? You're going to say, oh, I should worry about confounds. How am I dealing with that? Either as I estimate the data or after I get the data out and do my analysis. How do I measure each connection? It's going to turn out, and you'll see in functional connectivity, you have many, many options. I'm going to give you a little bit of a handle on the meaning in structure, as I will try to do also in function, so that your interpretations are potentially stronger, or at least point to new things you can ask, right? How do I account for pathological confounds versus features of interest? It is not trivial in diffusion imaging that the algorithms are not perfect, neither is the imaging, as we'll see in a second. So like, what do we do to add some constraints to help make sure we're not just looking at gobbledygook and false positives? So I'll mention something about that. How concerned should I be about signal to noise, reliability, validity, replicability of the findings, right? So we'll talk a little bit about that. And do I tell everybody everything I did, or only what I thought the reviewers would find interesting? That's a whole nother conversation, which we can have another day. I just think that that is a common problem in this space. So again, I'm going to encourage you just go back to Porvo's overview and then directly to Cieslak's paper, which is a beautiful piece of work about how they've tried to tackle a couple of problems all at once. How do you define regions? Well, their solution is, let's just take every parcellation people have on offer that is cited above some minimal count, and we're going to just run them all. And I'll tell you a little bit about what that means in a second. But in a nutshell, what you're doing is you're getting little voxel estimates about where directionality might be going. You're going to get some estimate about, with those directions, how can I make tracts? And if you're doing a connectivity analysis, you're probably picking a parcellation these days. You're going to say, how am I going to carve up the brain to build out my network? So you may know, but I want to point out, it's worth mentioning every time, that when you're doing diffusion tractography, again, everything is inferential. You've got a three-dimensional volume that you estimated, right, from the interaction of the MR field with this meat wad we call a brain. And then once we have those data out, we're going to have options for how we decide things at the voxel level. And so in general, what we're trying to do is say, I believe the corpus callosum connects, for instance, the inferior frontal gyrus on both hemispheres. I tend to think it does, probably. So let me go kind of construct a tract between those two. So the way that the algorithm will go about that is, at each voxel, we might have a principal direction that it estimates. You can't get under the voxel. That's what a voxel is. So you're always kind of estimating at the resolution of your data. And you can see here that these little ODF fields have little estimates for what direction it might be going, right? Now, over time, people have built competitor softwares and philosophies for how you treat certain details of this process. So this, in my view, is one of the first places where people's eyes gloss over and they stop getting interested or overwhelmed, right? I'm going to tell you that the good news is Cieslak and colleagues basically just said, let's build a pipeline to just estimate it all. They kind of point out which packages are cross-compatible. You can see that in that paper. But the good news for the investigator is it's not like people are talking totally different languages when they do this game across softwares. You're going to notice like the things pointing this way at the MT, CSD, MR-TRIX3 procedure are pretty similar to the MAPL procedure, to the GQI procedure, and so on. So what's interesting is this little quadrant down here. Now, these little blobs, you might realize, are off the corpus callosum in this little image. And those tend to be situations that involve crossing fibers. So the problem is that in the brain, things are free to go in multiple directions at very small adjacent areas. And so what's going to happen is we're going to start to have this second problem of estimating what happens if it's crossing over. How precisely can I estimate that, right? So that here in the MR-Trix procedure, we're going to try to tune a precise estimate. That's why those X's are narrow. In other softwares, they're a little more probabilistic and a little bit more broad. There's a little more tolerance for error. And you can see that these really blobby looking ones are kind of more indeterminate, right? There's probably a cross in there. Maybe there's some kind of pathology in there. It might be hard to know the difference. So these things all contribute to what happens when you built your connectome out of these data. So the advantage to the CSLAC in colleague style approach is let me not assume what I don't have, which is actual ground truth, because if we had it, we wouldn't be doing the imaging. And let me compute the properties in mass, and then I can do a lot of things with that. So let me just talk through that briefly. So this is just to point out that in even the same problem, if you were to measure from different connectomes, these methods are slightly sensitive to the number of directions you have. They're slightly sensitive to if you have a single or multi-shell technique. They're slightly sensitive to if you want to use diffusion kurtosis imaging to filter it first, right? So using something that a big group has designed to give you lots of possibilities is one brute force way to deal with that, is to say, let me not have too strong a prior on the best way to do it without evidence. Let me compute it multiple ways, just a philosophy for that. You should also know that once you have that imaging, your next decision is, well, do I want to say that the fibers just go from voxel to voxel, or do I want to say there's some chance involved, right? And so people have invented various ways of deterministic tractography, which is basically literally just track, oh, this little X here is on the left is pointing a little more left than right. Let me draw a fiber directly through the points of that X. Probabilistic is going to follow the shape of that and say, well, like maybe I'm going to trace a fiber there and a fiber there, and we're going to see where they go. And then we're going to try to like cut out things that are likely to be noise after the fact. That's roughly speaking the difference between the two. When you've played this game, I think still the most comprehensive treatment of this, I remember at Moss talking about this even a couple of years ago, but Meyer-Hahn did a very meticulous study with many groups to test these pipelines and anchor the analyses to very specific tract structures in the brain. And so they had this nice case where they had ground truth objects they were looking for. And in a nutshell, what you can see here on the graph on the left is basically you can overreach how many fibers or overlap. And so you want to overlap more than you overreach. And there are some things that are harder and easier to find on that landscape, right? And so the things that are kind of right here in the middle are pretty hard to differentiate in terms of like the structure versus the noise, basically. And if you see on the right here, these are example structures. Some of them, like the corticospinal tract, are really poorly pulled out from things like DTI deterministic tractography, pointing out here, down here. So we know there should be this nice fanning structure to the corticospinal tract. This is quite impoverished. If I used a probabilistic sequence, it actually fills it in quite nicely, but then there are also a lot of false positive fibers to that. I'm going to talk about what that means in connectomes in a second. So I do want to say to people that there has been progress here. So I think sometimes when I look at this method, I'm tempted, like I want to throw it out some days and sort of like it's a little bit hard to figure out where the truth is. But when you look at some of these controlled studies for things like the corticospinal tract, there is progress. So here now 14 years ago, there's an example of the descending arms of the corticospinal tract. And what they would have liked to see in this paper, and they drew the red lines on themselves, was where's all the branching arborization in these other tissues? So they knew right away, like, you know, our algorithms are privileging this big principal downturn and not the other stuff. Well, long story short, with these higher sampling rates, with more probabilistic techniques, with more filtering, we're doing a good job filling things in, I guess is a fair way to say it. You've got some false positives here, like I've mentioned, but if you kind of know you want to get the mass of a structure and tolerate some imprecision in the exact tracks, we can do it, right? A lot of these algorithms actually work quite well under that constraint. And the other nice thing about it is if you look at DTI or CFARI, which is another variant, DSI could be on this slide. If you have a lesion here on the right and you watch for things like progression in the lesion on the background image, you can also see sensitivity and lost connections as the tractography has gotten better. So here in the bottom row, you can see some of these finer branches to the corticospinal tract, and we have gotten more sensitive to effects like that in general with tractography. So that's a kind of progress. So last thing I'll say here, and I'll take a pause to breathe and encourage questions, is I don't know that you need to do a network question in the way you might have thought about it. You might have a connectivity question that is still about a single tract or a part of a single tract, right? And maybe if you're sitting in the language domain, you realize that what I need to know is about the arcuate, and I want to seed based on some part of the cortex, and I want to get that tract out, and I'm going to look at that tract. And that is a completely coherent and encouraged way to do that analysis up to this day and in the future. And so when you have the case where you can kind of fill in the structure and tolerate a little noise, go for it. Go ahead, region seed it. Maybe you know that you're interested in the association between the corticospinal tract and the superior longitudinal fasciculus or certain branches of those trees. Find a way to build a seed, pull out that tract, use an atlas. That will get you a long way for your hypothesis-driven questions about those tracts, okay? And so you can constrain pass-throughs. If you know that it needs to go through a certain white matter branch of the brain, well, you can build a little circle around that and make sure your fibers only traverse that and look at that and so on. And so again, there's nothing wrong with doing this. And I would say, frankly, a lot of us got really excited about the network stuff and forgot we asked the basic questions in this domain, certainly of which there are still many in my view. So finishing up this arm of the module, why are we still using this stuff when it has certain problems? Well, it's still the main option, right? So if you're not going in or going post-mortem, this is a way to get estimates for these structures. It does have descriptive and predictive validity in certain healthy and clinical samples for certain outcomes. And it allows for tests and theories and proofs of principle and network analysis while we're waiting for yet better technology. So I'm going to take a breath there and pause because I just did a lot in a short period of time and see if there are any questions. Going once, going twice. All right, this is the fun part coming up anyway. So all right, so let's say that you're done with tracts, or you say the tract is part of my question. Let me talk about your data elements very briefly. So I'm not going to go through or read all of these, but in general, there is established evidence that certain ways you can define measures on the tracts are related to certain kinds of structures and pathology. I should say that across these data, and I try to kind of do an update every 6 months to a year myself on this, there's rarely a thing that tells you like this specific thing about my axon or this specific thing about my cell body. But there are correlated trends that are more sensitive for some measures than others. So the way we've typically organized this is measures like fractional anisotropy, which is just kind of like the principal directionality of the tract, are nice omnibus measures. It's like they're kind of sensitive to a lot, but not very specific. And then you can further break a network down if you want to try to kind of get at more specific anatomical hypotheses, some of which are mentioned here. We usually also look at streamlines as a companion to that because it's sort of like, one is like sort of how directed and robust is it? The other is how much of it is there. So just kind of counting up the streamlines is the mass of that tract. All of those elements go into a matrix like that on the top right of my screen, and they all get weights, should you so choose, right? So how strongly connected are these things according to this measure? And again, in my view, there's a wide open terrain for the most part still for most of these measures and most samples most of the time. There are simply many more opportunities to ask questions than people asking those questions. So again, happy hunting ground here. So just because certain people started in a certain place doesn't mean it's not meaningful to ask physiologically specific questions on top of that. And so again, I think that when you look at the image here on the bottom left, you're computing this object, this tractogram. You might care about a circuit. So here you can find beautiful arcuate fasciculi in many patients, right? What we'll talk about when you build a network is sometimes that network parcellation is kind of violating what you wanted to look at. Other times it's finding you lots of what you want to look at. So we need to kind of think about that trade-off when we start defining them. Yeah, please. Yeah.
Are all these measures available with whatever kind of tract estimation technology you're using?
So every major software I'm familiar with, well, yeah, you will at least have the option to export this value for every tract you extract. Yeah, and so I think in the QSI prep philosophy, or even when we were doing our own custom pipelines back in the day, I would just like, even if I knew I was just looking at one, data storage is pretty cheap these days, just dump them all out. And then we kind of start with the main measure. And then if we have an interesting follow-up, you can go down that path. Yeah.
Are there interactions between the track estimation methods that you use and the validity of these measures that you pull out?
Very good question.
Are there interactions?
So yeah, thank you. So the question was like, is there any kind of interaction between your tractography method and the validity of these individual measures, right? Sort of, right? So if you think back to that picture of the crossing fibers and the estimation, right? So you're going to have kind of different weights on different properties of that voxel. And if I kind of believe like, well, I'll just use radial diffusivity as an example. So radial diffusivity is how things are moving across the axon, you can think about it. And so if you damage or demyelinate an axon, you have opportunity for less structure and more flow of fluids in that direction. Some of these methods are going to kind of like tune up, more precision in the direction in one dimension than the other. So like they'll be a little more sensitive to the axial diffusivity than the radial or vice versa. The weighting of FA mathematically is typically the same. So it's more just sort of like, how do you weight the lower level estimation of the data? Yeah. And I don't know how honestly well explored that is comprehensively. Like I imagine even just looking at those properties in individual syndromes probably has interesting information, would be my guess. Yeah.
So this seems to me like a good example of a place where the neuroscientist would be able to talk to a network scientist, and this would be an example of
Correct. So what he said, and this is a brilliant thing to point out, is like, this is a great example where that level of conversation is a domain scientist and network scientist could have a meaningful conversation, right? Because the network scientist might be really familiar with running FA networks till the cows come home. You might come on and say, look, I'm looking at MS. There's all this evidence about mean diffusivity findings. Like, maybe we can have a smarter connectome for that specific problem. And you can talk that through. Great point. So there's a good model for that, is like, start thinking about those things if you want to do structural connectivity. What do you know about the physiology? Is there even kind of like a correlative relationship with measures like this? Maybe that will influence where you start, right? And again, the nice thing is you can get them all at this point. So if you just want to get piles of data and treat some of it as rainy day, good for you. I need to talk about the problem with parcellations. It's a problem. It causes me consternation. I'm going to try to simplify an attack strategy for you, which I think usually holds up to most cases. Don't try to read the details on here. The point of me showing you all of these images is QSI Prep will be a masterclass in dredging out connectomes from all the parcellations here on the top left. And we've added a couple older ones for internal consistency for our stroke sample. And then on the top right, you'll see across the columns, there's a whole bunch of matrices here. That's what the thick lines are. But each one of these matrices contains different data elements from the same subject. So this is a whatever dimension by whatever dimension connectome view of a single participant. That's important because I can get up here on the counts. I can get, do I only count things that connect end to end? Do I only count them if they pass through each other? Do I get the FA measure? Do I get the mean length of those? And so on down that dimension. And then here on the y-axis is all the parcellations, right? So we now use this in our lab. We get like this kind of, pancake stack in multiple dimensions. And you can think of these as lenses through that particular brain. I would say in general, to save yourself some sanity, if you're kind of, bridging this domain scientist to connectome process, go back to what you think you know about your syndrome. So if I'm a language person, and if I can't ignore Broca's and Wernicke's, even if I have some problems with the model, probably you have some thoughts about, well, what if I bisect Broca's area more superior or inferior? Is there good evidence that I should do that? Am I interested in the difference between Brodmann's 44 and 45? Is there cytoarchitectonic evidence that I should, and so on? Now, I can tell you that many of these parcellations, often based on fMRI, are not going to care about your presumptions about the way that works. Now, it might be related to them, but it might not be principled on them. So when you first start thinking about parcellations, if you know or think you know something about a circuit you want to test, use that as a filter, use that as a question, your parcellations, right? That's an anchor point for you to say, well, cool, I can run that network analysis, but I might, before I even run it, gonna have trouble knowing what it means because I can't connect it to that property of the circuit. I think that's a very strong litmus test for most connectivity questions we encounter. So generally, you can kind of take one or the other or both of these strategies. You can pick a parcellation that you think already expresses the features of interest you care about, your subdivision of some anatomy, its connections, or you can test the concept in several parcellations and model their effects. I think you're more likely to do the first thing the more you think you have good domain knowledge and definition of a circuit, which I think we should always want. And I think you're more likely to do the second thing when you know a little bit less about that, but you suspect that there's some kind of organization to these parcellations that matters to you. My personal favorite example is there's now so much evidence in the frontoparietal case, including individual subjects, that part of our last RR1 was rejected because people kind of already had swallowed the pill on part of that story. And so there we can leverage, all right, people are pretty convinced, including our own data, we can use it for X, Y, and Z. What's the next step there? In other cases, I would say the language domain is my favorite example. These don't express a language network, so to speak, at all. You probably, if you study language, think about specific maps. They might be lateralized. They're encompassing the Parasylvian regime, and you're just not going to see it in these parcellations. I think that that's a scientific problem. And so I think you have to kind of have these interactions within domain and start to figure out how to sharpen up the questions, which is not trivial. And I think that we need a lot of work there. Yeah.
So, John.
Parcellations, what do you mean?
Let me Branch, I'm going to take Brian's question. And yeah. So say that one more time, Brian.
You say that you're just not going to see it, the language network in your parcellations.
Yeah, good question. Yeah, let me be very specific. So, and I'll use here, this is an anatomical AAL atlas, so old and based on the cytoarchitectonic and anatomical divisions. You won't get like a single color label for these are all language regions in any of these parcellations. You can see down here, like, you know, all this Parasylvian stuff is broken up into different real estate or modules. That's the problem. You just don't see certain networks you might think are a network in certain ways of parcellating the brain.
But then in other cases.
Yes, and then in other cases, so frontoparietal networks, I would say, are one of the more successful example cases of, you know, it looks like this is something that we can get our handle on and actually represent something about brain function at this point.
And sorry, I know we Branch had the question too.
Yeah.
Does that say, what does that imply?
To me, that implies that we're too preoccupied with fMRI, I guess is one thing I actually believe at this point. The second thing I think it says is that certain networks probably have like hierarchical privilege in certain methods. So like, it's probably not a coincidence that the FP frontoparietal story works better in a lot of these fMRI methods. But I'm not even sure we really understand why that is. And so I think, you know, when you have robust literatures like motor domain, aphasia, whatever, where it's like, we're pretty sure this stuff matters, you should kind of use your Bayesian prior and say, like, maybe I need to care about that more before I start analyzing. And that might be the hardest part of your project. I feel like that a lot at LCNS, for instance, where it's just sort of like, increasingly every time I start a project, I'm like, man, like just defining that first step is very difficult for certain questions. Yeah. Branch, go ahead.
So why, you alluded to something which I actually didn't know about these parcellations, how they were generated, and apparently it's fMRI data. Maybe you could speak a little bit more to that. Why isn't there an explicitly anatomic microscopic based parcellation? So the AAL sort of gets at that, but if you look at the AAL, I mean, they don't distinguish BA 17, 18, 19 in the visual area, so it doesn't really correspond. I mean, what the hell? I mean, this goes back 100 years. Why not have something that's based on what has been the defining nomenclature and organization for 100 years? Why come along and ignore that?
I'm going to go ahead and be kind to the whole field and say, like, I think it's a hard problem, but the specific reason it is hard is because when you actually start imaging the brain, we don't have any like really easy off the shelf implementable imaging that instantly gets you things like cytoarchitectonic regimes. There was the David Van Essen effort at that to do some sort of like myelin and white matter mapping. It does require certain specialized sequences. And I've never been able to figure out if the reason it hasn't taken off more is because it's hard and kind of specialized, or if people were kind of skeptical about even the principle that was published on. So I think that's probably a fair summary of where we still are. And I think people do use fMRI postulations a lot, probably dominantly, because of this history of first getting there and then clustering things up functionally And then there were some early successes with like default mode and frontal parietal that associated with other extrinsic variables. But then people generalized that probably a little bit past its skis for certain questions. And then it became like the dominant way to cut brains up. And then people do it different ways, and some are individual and some are group, and then some like privilege other pieces of information, but that's the lion's share of how we still do it. And I agree with you. This is now as I get crankier in my early mid career, I mean, you know, we need something better. Like it's obvious every time I go back and teach my undergrads in neuropsych, I'm like, I don't think Brodmann was wrong. So why can't we do a better job at that more than 100 years later? I don't know.
Thanks.
I know that's not satisfying. I'm dissatisfied too.
No, it is what it is. I mean, that's just the way people have done it. I don't think there's anything magic about Brodmann, but I do think he spent years and years and years looking at the brain under the microscope at, you know, hundreds of people, and came up with something that's been pretty valuable, I think. It just strikes me as odd that it would be ignored by subsequent investigators.
I still wonder, you know, we've talked about 7T on and off. I mean, I wonder how much of that is just like, maybe we'll get to a resolution and have some clever algorithms to cut it up a little better soon. Or maybe we don't quite have the imaging technique that's going to let us do that robustly. I'm worried it's the second thing.
Yeah.
Yeah.
Related question. To what degree have there been attempts to do network science using group parcellations like Brodmann, or I believe the Lausanne is also anatomical? And so to what degree have those been used in network science, even though the parcellations at the group level fit in?
Good question. So how have the sort of group level anatomical parcellations we can talk about been used in structural network or other network science? I would say in general, they've kind of fallen out of vogue, right? So I think people once they started seeing, well, I can get higher resolution, many regions with things like fMRI, large samples. They kind of figured, well, I can always go back to the anatomy if I want to, but this fMRI adds another layer of information I can use to cut the brain up. So I think that's caused kind of a communication issue where, I mean, probably we're familiar with this in LCNS all the time, where like I get more neuroanatomical the older I get, but the more functional it seems like the literature tends to get. And so to me, it feels like there's kind of like this bigger tension over time between the kind of basic organizing principles like Branch might be most familiar with, and the way people computationally cut the brain up. The problem that's caused is now we have all these parcellations. I'm working on this report right now. We're trying to do simulations for stroke on the big modules in the brain and across parcellations. It's really hard to reconcile sometimes the differences because there's like no organizing principle that connects them other than the fact that it was a brain and it was parcellated. So I personally think this is a much bigger problem than it's given credit for still.
John, I have a question about the parcellations. So I know I'm not very familiar with all the ones that are out there, but just looking at the ones you have up there now, they really seem to be very cortically focused. I was wondering if like if you're interested in questions about cortical or cerebellar structure or something like that, are there parcellations available to help you do that?
Yeah, so something we had some success doing, and I'll say this back, it's sort of like, what if you care about more than just the cortex, basically? And so, you know, if we go in and look at the thalamus and other structures, we have all these subnuclei. There are a handful of pretty good kind of group MNI atlases for microstructures. I think thalamus has its own atlas. There's, I think, one for other regions in the basal ganglia. I think the Dietrichson atlas in its revisions is probably still the best on the cerebellum. So, you know, one of the problems at Hello, and I knew this almost as soon as I started doing network analysis, is you're kind of like massively undersampling the complexity of those subcortical structures almost as soon as you start. Because if you kind of imagine like the imaging, as soon as you start trying to do diffusion imaging on the thalamus, well, every voxel is enormous compared to the thalamus and its nuclei, basically. So getting kind of the precision over these really fine pathways, I think, is still a pretty big problem. I think the highest resolution imaging that's kind of specialized at those regions looks, to me at least, like more successful at this problem. And having some atlas in there is probably better than no atlas. So we've usually tried to at least plug in like macro subcortical structures and a cerebellum atlas when we're trying to do Houblon analysis. Because it changes the measures by definition, right? I think getting under, just to close that diatribe, like the subcortical problem, again, it's a huge area of need and opportunity in my view. We know so many things in the brain depend on those cortical-subcortical interactions or subcortical interactions, and we're not doing a great job measuring them in most cases. We know they matter. And I think our connectome science would benefit and look better. It has to, right? All right, I'm gonna try to, we're having good conversation, so I'll try to be as brief as I can while being informative, but I really insist people interrupt here. So what I'm gonna do is just give you a couple example projects, either published or wrapping up. And these are in large part with data and collaborations with Roy Hamilton, Branch, who is on the call, Coslet, and Peter Turkletop at Georgetown. And so we've really benefited from these data and colleagues. And, you know, I've been thinking a lot this past couple of weeks about what did we really learn from each of these projects and how could better questions have helped? And I think, you know, when we consider this problem of what we're measuring, everybody who hasn't done network analysis yet should be aware of the Brain Connectivity Toolbox, still curated by Olaf Sporn's group. It's a tremendous resource if you just want to kind of go in, take some stock measures and start looking at a brain. But how do you navigate the choices? Because I was thinking about, I could show you the list of dozens and dozens of measures. You can invent your own today, right? So when I'm confronted with this problem with colleagues like these about, well, what's the language circuit and where do we go next? What I see is the problem on the right, but as this big diffuse network disruption. And the problem is, almost every time I've tried to ask a question about that, it is incredibly hard to get back to the thing on the left. So what is the disconnect conceptually between the 100 years model of neuroscience and the sort of things I think are novel, new features we can find, but like where's the mechanism? What's the model? So that's the attitude I'll take to our own work here. So here's one of my happiest examples of being past your skis and then realizing you needed to kind of strap the boots on a little bit better. And this is my fault, not Harrison's. Harrison is doing great work. So we're recently now going to need to submit again a recently rejected paper extending some work we've been doing for a few years, where I've been interested in the following with a couple of colleagues. We look at language function, and we tend to think of this sort of Broca's-like IFG area and its subregions as related to production in language and outputs. And there's models about what part's semantic and phonological, and every piece of that is a tricky story. But usually if you're looking at outcomes, IFG has something to do with it. If you just do kind of gray matter or functional mapping, the more preserved that left IFG is, the language outcome will tend to be better and it's most frequently implicated. It's also interesting that if you go to the other hemisphere, you notice that this node is quite active, this little IFG node, after the left IFG is damaged. So that homotope is maybe working overtime, the theory goes. Maybe we need to inhibit it. Maybe it'll get better with TMS. Okay, so this is kind of a classic now, couple decades story. And I think there's a lot to it. We can hang our hat on that. When I look at this problem, the first thing I see is, well, I've got two hemispheres. Maybe I'm going to damage this thing on the left. And immediately I see more than that thing just damaged on the left. That thing used to be connected to all of these nearby neighbors. Those nearby neighbors were part of bigger paths, and so on, all across this hemisphere. Maybe that was damaged within a kind of big connected module, or maybe this was a bridge between modules. All of those things might matter to me in terms of how that system functions. Kind of even worse, then I move to the right hemisphere, and something that I don't think occurs to us often definitely happens in the data, which is as soon as I blow out something on the left, by definition, you think of the whole network as a unit, the role a node takes over here will change. Maybe a little bit, maybe not much, or maybe drastically, depending on its position in the network in that person. So Harrison and I have been talking about this for a little while, and we're trying to kind of like connect between the recent work we've done that's a little haphazard back to this IFG story. So here's one version of that, and then I'll tell you what I think it does and doesn't mean. So a few years ago, I was working with Danny Bassett and had this thought that, well, controlling networks is interesting. I like engineering techniques. I want to try to fix it by controlling it to a better place. Network control theory has measures. One measure is boundary controllability, and that is this kind of intermodular measure. So it's high when a node is sitting between lots of modules, and it's low when it's kind of like a little micromanager nearby, right? So the thinking there was, well, the more a node has to do in the kind of big modular structure of the network, might, on the one hand, give it advantages, but on the other hand, burden it. So we had a couple bits of evidence from healthy data that, kind of the bigger role this node has in the left IFG, the more of a problem it is for managing certain high semantic demands. And we kind of had a sort of, hand wavy, maybe it's a noisy node that's buffering too much discussion. So we wanted to try to take that concept and say, all right, well, what if we put that over in stroke and on the right side of the brain? So keep in mind, this is about semantics on the left and health. We're trying to move that over to the right in stroke and aphasia. So the first thing that we can kind of take to the bank, which was quite cool, is that when you damage the left hemisphere, compared to neurotypical people at right pars triangularis, where a lot of the kind of TMS is aimed for these studies, very clearly in persons with aphasia, the role of that node in that intermodular process goes up. So this is either super obvious and issue or to me kind of profound, because what I just told you is that I blew the thing out on the left, and in the structure, I had a profound increase in that role on the right. The structure. I didn't do any functional imaging. I just am implying by virtue of this measure that this outsized role has increased on the right side, right? So the idea there is if that node is occupying more of this role, my first guess was cool. Well, if this would be super convenient, the right side is going up in that role. Maybe that's more of a semantic interference on that. Maybe they're going to do worse at semantics, and that would be a very tidy story. Well, of course, nothing goes that straightforwardly. So we did find something which is kind of interesting. What actually happens is that the stronger that effect is in that node on the right side, the better people are at the phonological processing. So I'm going to tell you a couple things about that. The first thing I'm going to say is I bet the parcellation has something to do with it, because the way I cut up this network in the first place has a lot to do with its values. This is the strength of kind of either picking a better parcellation or scanning across a couple. So that's part of what we're starting to look at now. The other thing, but I think this is the insidious thing for neuroscience, is the following. So let's work backwards. I just told you it's a region and the other region occupies the role, and I can make it better with TMS. I wish everything was this simple, right? Because I would just go ahead and bang on everything with TMS and be right 100% of the time. And we get all of the NIH grants and never have any stress again. And all the problems would be cured and I'd be out of career and I'd go retire, right? Awesome. It's not like that, unfortunately. So what we first have to ask in network science, which did after the fact, was, to be honest with you, was, okay, let's hold our horses. I have to get back to that left IFG. So the first thing I need to ask about is, wait a second, before I even worry about your role and everything else, what about your role with your neighbors? This is the number one thing. I want you to look for this in network studies. This is the number one thing most people skip in most network studies. They do not tend to look at the network measure they're interested in and then compare it to the literally simpler measure mathematically. So here's a case where one thing considers all of the structure and complexity in the network compared to the most basic connectivity of that node. So just look for whether they control node degree or strength in their node analysis, for instance. So that didn't account for the effect. So that's kind of exciting. We're saying, all right, well, this bigger intermodular thing says more than just the local connectivity, right? So, I'm kind of leaning toward maybe the role in the bigger intermodular network. There's something to that story. We're not alone in that evidence and point in general. This is just the bit of evidence from this specific measure. But here's what's really haunting me now. If I just continue to strip away the complexity from this story, I get back to basically what neuroscience has been doing for 100 years. And I say, wait a second, what if the only thing that matters is the connection between that node and the thing on the other side? So this would be sort of sheepishly, the no duh, you would probably start out with a project if you weren't excited about the networks. You would come back and you'd say either the mediating fiber for the corpus callosum or the arcuate fasciculus to the region I care about from my language model might matter more. So of course now Harrison's doing a bit of follow-up to that with Branch. We're kind of seeing that explain some of the phonology on the right hemisphere. So what's my advice here? Don't forget about the neuroscience. Start with the simple scenario. If you suspect the mechanisms are more complex than that, and they probably are for most things, you still need kind of a stepwise logic to understanding it. I use the car metaphor shamelessly now. If you were going to tell me, John, go build this car, I wouldn't just look at it and say, I got it. I don't have the knowledge to do that. I'd probably need a lot of training and some kind of articulate instruction manual and a team of people, right? We need to probably think about brains like that when we're doing these little dismantling studies. If I am connecting the axle between wheel and wheel and I realize it needs a drive shaft, I start asking that next network question. But I still need the wheels and the axle to support the car. And so I think that, this is, again, a out loud admission that, we got to back up and ask these questions to move forward. Let me pause there and ask any questions. Okay. Oh, there's my questions. All right. So another exciting piece of work is Brian Erickson had worked out a couple of years ago with Peter Turkeltaub's data, working on this problem of bypasses in the brain. So something that I think is really interesting, and there's a bit of evidence from other groups, is, again, when you damage any one thing in the brain, there's a lot of paths to begin with. And it could be that the brain uses a lot of the paths that are left to get back to healthier functioning or kind of robustness, or causing interference in that brain as you overburden certain different roadways to one extent or another. So here we're talking about paths, like on the bottom left, where what we're going to do is add steps along this path, right? So we're going to start with the assumption that there's something like here, a stroke. We've knocked out an identifiable set of regions. Once we've done that, well, we can lose some nodes and some connections. And when we lose those nodes and connections, we have many interesting scenarios, but the couple we were interested in here is, well, you can knock out the connection, just shear through the white matter, but the region's preserved. That would be node 4 here. So node 4 happily would have communicated with node 1 before, and it no longer can. So if it wants to communicate with node 1, we might need to look elsewhere for how it paths around that lesion, okay? The other thing that happens is, I knocked out the node. The gray matter is blown out. Well, I think of that mechanically as just gray matter cell bodies connected to white matter axons. The whole party is now crashing down. And so when we blow out that node, we have an additional problem of multiple links going at once around that node. Typically, there's an issue of degree, of course. What that looks like in a matrix is the stroke gets worse and worse. These bluer and bluer areas are basically showing you this is just massive connection loss in that stroke sample. But there's a lot left. So what can we ask about how the brain might structurally reroute information through that network? That's the question here. So let me come back to reliability and validity. I would argue you rarely have validity. You don't know about validity in the sense of physiology in most of these diffusion methods, unless you had post-mortem, tracer, other studies for that problem. You can think about reliability. And so in reliability, we were saying, all right, well, we want to know that when we're asking something about bypasses, we're pretty confident that they were there in the healthy sample. I still am fascinated by individual differences. I don't think any one of our two brains is exactly wired up with the same paths. It's probably got the same big stuff, but the little stuff probably varies in very meaningful ways. And there's a lot of network science to suggest that that's meaningful. What we wanted to start was to say, let's make sure everybody has this set of connections, so we understand among what's available to everybody before they have stroke, what bypasses they could use. So I'm just kind of saying that that's an interesting conjecture we should talk more about. So we get the matrix on the right. That's basically saying these are the edges that are connected for everybody. Now let's search in that space. So when we do that, we can basically pull out a couple of ideas. We can say, all right, well, if I need to get from Broca's to somewhere around Wernicke's area, I might be able to get back there in a couple hops. Maybe I knocked out a direct connection and I can look for the shortest path there. And I can give that some weight as a function of the structural weights in that matrix. I can ask about elongations. So before, this might have been a 4-node process. And then here, we're going to add some weight and length to that. So how much longer did the path get from node to node? It's got farther to go. It's not as efficient. This is an interesting concept to me that I think was kind of understudied and still is. This concept of overlaps kind of fascinates me still, where before, what this brain was doing is, here's the roadways. So many people are getting from New York to Philadelphia. Awesome, right? The road is going to, back up to this degree and the traffic is predictable. I blow out, I-95 corridor, right? Now, if you've ever driven on that road at any period in time, this is a disaster for driving between those two cities. The problem is another one of these weird, what I call simultaneity problems. It's not just that you blew out 95 and you had to reroute the traffic. It's that every other thing that was going on implies overlapping demands on that new structure. In other words, don't treat it like the old structure that involved 95. Treat it like a new structure that doesn't have a 95. And when you think about it that way, as the brain has to adapt to that, you have all of these opportunities for competition for the remaining resources, the pathways. So we computed some measures, Brian computed measures about, all right, let's find these paths, let's figure out how long they are, and let's find where they overlap. And now let's use that to try and predict behavior. So one of the interesting things to come out from this is that all of these strokes were on the left hemisphere in aphasia. And what we found when we did an analysis of hubs, those are the things that are kind of mediating short paths on average in health. Like, are those hubs eating up what it reroutes to, right? So the intuition there is, I already had a pretty important role. Do I become even more important once I start blowing stuff out? And the answer to that is a little bit more complicated, but kind of intuitive. And so if you have a higher hub status, the average elongation, tends to be a little bit lower, right? Paths are shorter, right? We're relying on the hub status to kind of keep the paths short. So that's kind of an advantage on average. On the right here, we have a hub status versus the bypass overlap. So in general, kind of intuitively, if you were already involved in short paths, you're going to gobble up a lot of the new short paths. This is kind of sometimes called the rich club organization or backbone of a brain network, where there are a few things absorbing a lot of the structural connections and paths through that network. So there's a potential for a lot of relays through efficient paths. And on the other hand, there's competition for those resources. Those are the two ideas that we're after here. So I'm gonna kind of blitz through this just because we don't have a ton of time. And the whole point of the blitzing through it is, this project, in my opinion, is a great example where we introduced a pretty cool concept for the network and measured it. And the problem is the constraints, right? So what we have here in front of us for just one of the factor scores from the WOB, lexical production, we can see that there's some center mass in the left hemisphere about where elongations matter for performance, where getting longer is worse for performance typically. And then we can kind of see where there's like positive and negative overlaps related to these reroutes. And so in general, we can have more overlaps inducing better performance and more overlaps introducing worse performance. And the idea there is that sometimes we're kind of teaming up with advantageous paths and maybe the traffic is gelling, and the other is maybe there's competition. Maybe this is overburdening the system. What we found that was interesting, speaking of subcortex, is that at least in this construct and another, we found that some of these bad overlaps heavily recruit subcortical pathways, the putamen specifically in this instance. So for lexical production, it could be that depending on how that stroke affects you, the more you're drawing in the putamen and subcortical structures, it's quite contraindicated for your outcome. On the other hand, if you kind of find a lot of overlaps and reroutes through that language network, it might go better for you, right? So we can find dissociations like this and mapping like this. For the sake of time, we could talk more about this later, but we found that there was some sensitivity in each subdomain. And we were able to kind of like visually map some of this back to the ventral and dorsal stream hypotheses for these systems. But in full admission, what I think we both found frustrating about this was it's sort of like, we knew even as we were doing it, like we're going to kind of get a spaghetti, right? We have this feeling that what's just interesting and frustrating about the brain is we're going to find a lot of sources of variances that are going to kind of sniff out with a sufficiently powerful method. I think the real work here is to come back, and Brian's doing an extension now to anchor it back to things like the IFG and for the production outcomes, and start to say, all right, can we find constrained reroutes, right, that help us navigate between this kind of old language model that persists and some of these modern methods for finding how the brain might navigate these grievous wounds with lots of complex changes. So that's where we're going with this one. Any questions on that? I know I went quickly.
Yeah, John, I got a question. This kind, I think, and I think Brian's on the call. That was great stuff, Brian. Really nice paper. But my question is this. So in fMRI, we've been doing some, not we, I don't do it, but people have been doing it for 25, 30 years, right? So, and the issue is it's a very, it's a technique that the signal to noise ratio is pretty crummy. So you need to do big studies, and the WashU people show that that the amount of data that you have dramatically determines the kinds of inferences and the quality of the results that you can get, right? So the more, the better in a big way. So the question that I have for this kind of thing is, do you get sufficiently reliable, reproducible, powerful results on an individual basis with these kinds of analyses, so that you could do something that I personally, with my predilection for individual case investigations, could study in this following sense. Say, so-and-so has a particular profile of aphasic impairment. Can I make sense of this on the basis of their particular pattern of connectivity?
Yeah.
Or this other kind of, there are other aspects of, say, the neurologic examination. You talk about the putamen, right? So you talk about, do people with this pattern of aphasic deficit consistently show deficits on the neurologic examination suggestive of impairment in the basal ganglia, for example?
Yep.
So to what extent can you take data from individuals and subject that to an analysis using a comparison to say a healthy control group?
I think it can be done. I think part of the answer is when you add constraints like what do we see reliable in a population, that was definitely an important filter. I think we actually went through a version of the project where we were like trying different less constrained variants, and you could make an individual differences case, but that seemed like it just had every sign of a noise contributing process. So I think these constraints on the healthy group can help. I think the second thing and kind of the opportunity here in the future and for us is once you know that you're looking for something like these properties and you have a person there that you're interested in, we can do things like find matched controls or subsets of people through that same connectome filter. You can also expect that because those tracks are supposed to be there, that if you also know just a little bit about the pathology, you can kind of seed the analysis a little more intelligently. I really felt like we both felt as we were coming back to say, well, like we're kind of fishing in this big network soup. The one thing we know we can come back to is anchor through the IFG. We know there are going to be a handful of dominant paths that are probably there before you had a stroke. And then we also know there's kind of a neighborhood around that in the network your brain could kind of search for to reroute things. My guess is that's going to work better in the long run because there's still so much, uncertainty in the like false positives, frankly, on the other structures that just gets noisier and noisier as you add more connections, is my fear. If you start in a neighborhood and kind of grow it out, there's probably more success. We had a project we started with a student of Peter's a few years ago that unfortunately he graduated and got too successful too quickly and it never got finished. But we did this kind of like local search procedure fitting to the data. It worked really well in individual subjects and we just haven't gotten back to it. I think if we anchor that to some of our intervention questions, it's probably worth revisiting at this point. So I hope I'm not just being too hopeful there, but I mean, I think the nice thing compared to some of those, like you need a gazillion fMRI subjects, is some part of it is really the tools that kind of exploit the signal to noise. I mean, we saw that in like fingerprinting analysis that was, popular with the IQ for a bit with phenidol. But I think if you look closer at most of those instances, the more prior you have on the feature selection just helps. And if you can do that with actual knowledge, that helps more. So I want to do it. I'm going to keep bothering you and Harrison about it. So sorry, go ahead.
No, sorry, I have a question. So like this idea about sort of finding alternative paths to the brain is a really interesting question, but to me, it feels like it sort of makes an assumption that the nodes that it's passing through are not doing any computation on the information that it's receiving, right? But it seems like that's probably not true because if you pass through it, if you get information sent to that area, it's probably going to compute something with it.
Yep.
And presumably whichever nodes along the way that you pass through are going to be doing different things. And so how do you distinguish between like this is a good choice because it's a short number of edges or nodes that you're passing through versus this thing maybe it only passes through one node, but that is a fundamentally bad computation to my data or to the information that it's receiving?
So first of all, great question. This is going to be like kind of the kind of cheeky under theme of the functional connectivity talk, where it's sort of like, if you're thinking a little more like Aaron's talking, you're looking at all these nodes and being like, well, they don't just like sit there idle on your graph. Like they're all doing internal computations, we tend to think. And I just got to say like that to me is like one of the top 3 biggest problems in the field in my humble opinion. It's just kind of like, I worry about this every day. Like to me, it's almost demotivating to do certain studies because it's just sort of like we do this study I kind of suspect what's going on with Harrison's data, for instance, is that not only is there like the homotopic potential for the right IFG to do the thing I thought it did, it's probably also computationally at that region, maybe managing something else. And I'm just not going to see that in the network data, not that these network data. So I do think people literally have designed network tools that sort of deal with this, called metadata tools, where you can kind of flag a node with properties. That's a potential area to look at, where if you have enough of a theoretical inclination to say, qualitatively, this node is interesting in this way, you can build that into a network analysis now. I don't know that I don't know that those are developed enough to satisfy what you're saying, though, frankly, right? Because you can kind of say like, well, I think this is kind of like an up node for this and a down node for that. But like really what you're talking about is like, this is the theory of the process of theory building for the brain. We need to keep filling it out for the nodes, and we're just not doing it. They're two literatures not talking to each other most of the time, unfortunately. Yeah.
So
Yeah.
relevant to that question. Really simple, but I'm not sure if I can be heard, is that we're looking here at an extremely simplified model of how a general property in the brain might work.
Yeah.
To support function after stroke, because we have trouble answering that question.
Yeah. Right. So what Brian said there was, we kind of had the feeling we were looking at this kind of model for bypasses and stroke as an option because we had trouble answering that question, right? It's sort of like we're all kind of trying to work around that elephant in the room when we motivate these analyses. Yeah. So yeah, I mean, for yourself, this is a problem. I'm interested in it. I think there's a lot of opportunity to keep asking questions there. All right, and for the sake of, I'll stay a few minutes over for the sake of completeness. If people have to go, I understand. I'm going to just talk very briefly about something we're following up on and trying to turn into an intervention opportunity to kind of enhance what we've done. So this is the paper I talk about where I did this with Peter Turkeltaub a couple of years ago, and it's an example of a project where we looked at each other on the Zoom call after, and we're like, he says, I'm not sure what question we answered. And I looked at him and I was like, yeah, I'm not entirely sure either. And so I thought about that for a while and how we got there. And that's part of what motivated my opening to this talk. And I did have questions, but I'm going to tell you what I liked about them and what I didn't. I did want to know this. I was kind of sitting there watching all of these connectivity studies and thinking about structure and worrying about questions like Aaron's and talking to Branch and Roy and Peter. And I'm like, I just don't even know how much I should expect to get out of these stroke connectomes. And it's a hard problem to open with putting a lesion in this complex network and then knowing what to ask next, frankly. And so to me, it felt sort of like, my postdoc exercise, and let me start to try to get into that problem and mechanically break it apart and then see what we can do. So that's really where this paper came from. And so I started wondering and worrying about this problem of like, a lot of the time, these poor people with stroke show up and like, they may never have gone through imaging before. Most of them certainly haven't. And like, you're just sort of like, here's a healthy person that may or may not be anything like this person you're looking at. Sure, you matched them on age and other things, but they're like, come on, like look in their actual brain. Is that the same kind of brain? So like, how do we grapple with individual differences, lack of premorbid data, and expectations, right? And I got really interested in this idea about like, is there kind of a way to tease out with a simulation what you'd kind of expect to happen from the control group? And then if that doesn't show up in the stroke group, that might be evidence of adaptation or secondary cascades in the network failure, things like this. So this was really, in my view, just a primer into that space. And basically what we're going to do, and I'll try to simplify this for brevity so people know what questions to ask, is we would say, all right, I can go grab a big control sample. So we had nice matched controls from Peter's lovely corpus, and we can start to populate estimates about what would happen if I start to stroke all of these healthy networks on a network dimension. And so that would be this yellow pile of observations. I have a whole bunch of healthy groups, and I can kind of randomize all of the healthy strokes, and then I can get out realistic strokes. And then finally, here's the actual stroke I observed. And you get a couple things here. First, you can see how much variance would explain on the outcome measure when I simulate that attack. And second, I can see where the actual estimate was in the stroke sample and kind of think of this as a confidence interval for where it may have been. And the difference between where this pole is, that actual observation, and this peak is is some kind of information about the difference between what is the stroke actually doing to my network relative to the healthy network. What's left to explain? So I'll walk you very briefly through what we did here. So with some clever tools from Dorian Pistina, we use this imputation procedure, which is now pretty successful in a few studies, where you have this problem of parcellations. Lo and behold, I wish we didn't have parcellations. They're so frustrating, right? But I'm trying to make a network, so we're going to use a parcellation. So we're using one of our anatomical parcellations. It's an older atlas called the Lausanne. The advantage to that was it's grounded in kind of major anatomical labels of gyri at least. And so we cut that up and we slap it on the subjects, and then we get a network. For the stroke subjects, the obvious problem is, there's a stroke there. So what happens to all the regions that were here? There's a couple ways you can deal with that. You can try to either modify the parcellation itself to try to make it adapt for that problem, or you can say, no, I kind of want to like have less bias in how I fit that parcellation, but there's a stroke there. How do I do it? So Dorian Piscina used a method where basically he kind of extracts the brain, mirrors information for that subject's other hemisphere, the intact hemisphere, uses some group average information and fills it in. And, you know, we don't really know what that brain looked like before we imaged it, but it usually gives you a pretty visibly darn good guess. Like it would fool me, and probably most neurologists we've had look at it, that it looks like, yeah, that's a reasonable brain scan. So this usually reduces the bias when we do the parcellation, and then we can compare these networks. All right, so what did we do? So the way I thought about this problem was, all right, well, I got stroke data and I've got some control data. And what I really want to know about this is if I kind of want to like have a lot more information about what to expect before I can see it, I want to take that stroke and I want to put it in as many observations as I can. I want to simulate that in every single control data in the data set and get kind of a confidence interval for like what's the expected value for these network effects. So we did that. We got a lesion mask for every single lesion. We applied it to every single control subject. So we had some 1,600 observations, right, of like simulated strokes in the sample. And once we have that information, we went along here and, sorry for the overlap, but we asked about 5 major properties. We wanted to just see how much network stuff is there. Turns out that's a handy proxy for lesion volume, but in a network sense. They're correlated, but not perfectly. So we looked at those properties. You can ask about how modular is the network. You can ask about the global efficiency, or how efficient are the paths through the network. Transitivity, not pictured here. You'd see a little highlighted triangle, right? These are the clusters. Small world propensity is a function of efficient paths and good clustering. And the idea here was, you're going to see this in stroke, and we're onto this project as the follow-up. Modularity gets a lot of fanfare. It's been associated with outcomes in some cases. The thing that really drives me crazy about not understanding is, well, the modularity changes because certain things break by definition. And the problem is sometimes it goes up and sometimes it goes down, and it kind of relates to outcomes, but it's harder to get the pattern there. As a simulation, what you can start to do is say, well, let me break each piece and kind of figure out causally in the network what pushes it up or down. That gives you a candidate pathway, right, for what's actually causing the shifts that you observe. It might be the whole lesion mass, and that's the point. It might be because you're bisecting certain important things, right? So this project is the first step along that pathway. There's a lot going on here. I'm going to pick one example and just point it out from the top left. So what we first wanted to know is that, well, when I take the whole brain or the lesion left hemisphere, I want to know that my strokes, so the whole stroke sample, and my simulations are kind of like approximating each other, right? I want to know that's a decent simulation. And this worked pretty well, actually. So we go in, we throw these lesions into the healthy people. Here's the stroke distribution. It matches the simulation quite well. The distributions are mostly similar in most of the cases, and they're mostly statistically different from the stroke sample. All right, so that's just kind of a validating step. In a nutshell, what we find across a number of clinically relevant behaviors is that if I look at the overall WAB-AQ, for instance, global aphasia functioning, what I should expect from a connectomic analysis is that in the real data explains something like 20%. And maybe we would expect it to range somewhere between 20% and all the way may up to nudging at 30% after controlling for lesion volume and a couple of other variables. So you can do this for each domain. You know, I wouldn't get too hung up, frankly, about sensitivity, specificity at this point, because these are such global measures. The point to me that I take away is there's a non-trivial amount of variance floating around, even in a really coarse analysis of the networks, after accounting for some of the major outcome predictors people look at in the imaging. potentially there's even more information in there than we got in a particular stroke sample. If these simulated attack lesions are proxies for what can happen in the bigger group. So this is kind of a sort of lens into, in more samples in the future, I'll kind of have my eye on, you know, are these estimates holding true? The other thing you can do here, and again, this is just for thoroughness, I'm going to focus you on the top left again. You can go back to your models that we used to predict. So that we used little multi-level regression models on these measures that we simulated. And you can see which ones account for the most variance. And so these are betas, and the betas across the simulation suggest that there's information in the whole brain network in things like the overall weight and how efficient the paths are. And those tend to be somewhat enhanced, unsurprisingly, in the left hemisphere. Now, kind of interesting here is, whereas people look at properties like small worldness as a big proxy for intelligence and other things, in these language domain networks for aphasia, it just really seemed to be like, how many connections are left and how efficient are those paths? Kind of get the lion's share of the weight for a lot of these measures. So to me, that's kind of good news, because it means that if you really focus on all of these other measures in their entirety, there's still some signal for some measures relative to others. I suspect, given that the literature keeps pulling out modularity, and we did find effects there, they tend to be a little smaller. What I would like to be doing in our samples now is continue to following up on this pathing question and node question, such as Aaron raised, and look also in the neighborhood and the bypasses around where Branch was raising. I do think that the overall simulation suggests that there really are properties across the connectome that we can get a handle on there. All right, so I'm wrapping up here. What are the open questions? Well, we talked about a lot. I still think one of the biggest long-run promises for connectivity analysis, including in structure, are these functions distributed or local, and to what extent? I think that network science in general has a lot of promise in helping you articulate questions anchored in maybe localizationist models, but entertaining distributivist hypotheses. I think that's a great opportunity for everyone. How does structure mediate function? I mean, both in the behavior and in functional signals. So I'll get back to that third thing in the third talk. But just testing that relationship is still not very frequently done. So I don't know how much of an outcome explained in a clinical sense that will get you every time, but theoretically, there's a lot to be said there. Can these algorithms be improved for tractography in the networks, or is there just a fundamental limit we're going to crash into? I think conceptually, one of them is what Aaron raised. Like, we're going to need to add stuff to the network into the story, period. I don't think there's any way around that. I think the algorithms, kind of back to Branch's question about individual differences, they're not done. I think we're going to keep seeing progress in the algorithms. I'm just worried we're going to need something better than diffusion imaging in the end for a lot of these. We're just going to. The fundamental property is not talking down on anyone. It's fantastic, but I don't know how we get underneath that. Maybe I can't see it, but it's tough. I do think the covariance problem, I give you hints of that with like, look, like decompose the complex thing into simpler things and pit them against each other. We need a lot more of that in network science, right? Mostly what you get is sort of, there's an exciting network association and it makes a splash for a bit. And it's like, first of all, not a lot of it reproduces, I can tell you, because we try sometimes and I talk to colleagues, right? But even when it does, you have the mapping problem, right? It's sort of like, well, why is that any better than like my single direct connection with the homotope? It might not be statistically, right? Maybe it's scientifically meaningful, maybe not. We have to think about it. And last, but far from least for me, how do we get interventions out of this concept? So we're doing a little bit of trial work over at LCNS, kind of thinking about it, interacting with brain stimulation. This is a huge frontier. There's a lot we can talk about there. All right, so I'm wrapped up. Any final questions before we break for today?
I had one thing just to say about
Yeah.
one of those open questions, which was about the reliability of the measures,
Yeah.
the appropriateness of the fundamental measures that go under. And that's just to say, from a more positive standpoint in graph theory and its application for science, that you're still dealing with those same measures, whether or not you're doing graph theoretical analysis.
Yes.
So you have the same reliability, validity issues when you're not thinking about the higher complex dimensional structure.
Yeah, so I mean, that's a great point. So I think, and kind of back to Branch's question about fMRI SNR, Brian Erickson is basically saying that you're going to have some version of these problems at bottom that you also see expressed in the network measures, right? You're still going to have these reliability and validity concerns in the more basic analyses. You know, it's possible that that kind of error inflates in some cases, the more the complexity analysis gets. But, you know, for most of these, you're kind of in the same ballpark. So in other words, like, I get pretty suspicious whenever I see, you know, the machine learning paper that's like, here's 90% of the variance with this imaging measure in that sample. It's like, almost certainly I can tell you that that is going to plummet the next time you do true out of sample data analysis. There are just too many ways for you to train to the error structure in a data set, no matter how robust the in-sample machine learning algorithm was, that are probably not going to help you in the new case. And so again, I think that that's just a case where, yeah, we need better methods. That's a general problem. And then I just, it's plain old fashioned thinking in priors. You've got to have good questions of your circuits and your clinical sample, and that's just going to help you in the long run. All right, my Quaker open mic, one more minute, if any other questions. And what I'll say if people are contemplating asking, so thank you for your time and attention so far. I'm hoping something was interesting there for you. The next talk, the way you'll hear me talk about functional connectivity is I'm going to give you a brief history of what is that and where it comes from. I'm going to talk about the kind of major categories of what people do when they try to make sort of causal versus descriptive inferences. I'm going to talk about dynamics in terms of what's up with this time varying stuff. Importantly, I'm going to talk a lot about physiology at the outset. And so when are we kind of in a terrain where this is like a pretty well-established principle of the signal, and where are we kind of like, we don't know if what's the heat and what's the mechanics, right, of the situation. So that's how I'll try to attack that conversation. Oh, I have one chat. Tiffany says, that was excellent. Thank you, John. You're very welcome. Thank you.
Thanks, John.
Cool. All right. Well, I'm not far away. If anybody needs to reach out, this was a pleasure. And, you know, I'm looking forward to chatting more in the next session, which I think we have, what, 2 Fridays from now, Porva? Because we're out next. Okay. Yeah. So 2 Fridays from now, same time, same format. And I appreciate everybody chiming in.
Thanks, John.
Yeah. Thanks, Branch. Thanks,
Thank
everybody.
you, John. It was great.
Thanks, everybody.
Bye.
Thank you, John.
Thank you.
Oh, and they're waving here. If you, yeah, they're very friendly over here. All right. See everybody soon.